Thank
Voilà.
you. Yes, I will do my presentation in English, but I'm happy to take any questions in French. So I'm very excited. Well, thank you for the invitation, Laurence. I've been traveling in France for a few weeks, and I'm very excited about some of the work that I've been presenting during this trip. And one piece of work we did is in one of our favorite topic, microbiome data analysis, and particularly for longitudinal studies. So this method, I hope, will be in mixomics in the next semester or so. But first, I just want to start with some introduction. So Laurence was mentioning that as a PhD student, I was part of the Biopus group. That was a long time ago now. And so now I'm directing a small interdisciplinary center called Melbourne Integrated Genomics at the University of Melbourne. And what is interesting about this group is that we are really at the interface between biology, mathematics, and statistics. So most of us are from the School of Math and Stats, but some of us are also from the School of Bioscience. And I think when I reflect back on, my trajectory as a researcher, the Biopus group was really, I think, in that same aspiration. And so I end up leading a group like this now. We're also part of two major centers of excellence that are funded by the Australian government. They are 7 years long programs on, really blue sky research. So I'm actually part of a center called QBIC, which is quantum biotechnology. And there I will move all my omics knowledge and all the work I've been doing towards more quantum computing for omics data. And the other center is called Maxis, and it's led by people in my center. And it's about cell systems modeling. So this, my center, Melbourne Integrative Genomics, is really involved in running computational workshops for biologists. We also have a seminar series and reading groups, so really hoping to create as many collaboration as we can with the biological sciences. And my group is a bit bigger than the picture now that is shown here, but we have expertise in statistics and computational biology. My team is mostly composed of statisticians, bioinformaticians, but also data analysts and software developers. And we really pride ourselves in multidisciplinary research. We're really interested in developing statistical methods that can be applied directly into biological problems. And the methods we've developed, such as those in mixomics, are often technology agnostic because they are non-parametric. And so I think this has really been the success of the methods in mixomics, is that they can be applied to a various range of omics data. Mixomics has also been recognized last year as a significant contribution to the software development for the biological community. So now I'm going to start with my presentation. Feel free, I will stop at the end of each section so that if you want to interrupt and ask questions, feel free to ask them then. So the outline of my presentation will first cover just characteristics of microbiome data, because I think it's important to understand the challenges we face when we analyze data. And then I will move on to the characteristics and the different types of biological question and data analytics question we can ask to longitudinal microbiome studies. And again, I will then mention all the challenges we face. And finally, the topic of, or the aim of this talk is to introduce you to our new method called LUPINE, which is proposing to infer networks for microbial communities. So I'll first start with microbiome data. The microbiome can be seen as an organism in itself. It's been enabled. So the microbiome is a collection of all microorganisms that we can measure in a given environment or ecosystem. It's been enabled by technological advances from sequencing and culture-independent techniques. I think it's been around for more than 15, 20 years now, but there are still a lot of challenges we face with those kind of studies. The idea of, or the aim of the microbiome is to characterize the composition, the interactions of the microbes. But it's not just about saying, oh, this is the organisms we have. It's also about understanding their biological function and how they affect the host. So most of the studies are on us. So how do they affect us, and how do they participate in disease? And so what we found out, and I've been working in that field for more than 10 years now, is that there were and there are still a lot of challenges from a bioinformatics perspective, but also a statistical perspective. So usually in a microbiome study, once you go beyond the description of the composition of your data in terms of these are the taxa we have, or these are the species we have, really we tend to be interested in the identification of the microbial features, so taxa, for example, whose relative abundance is associated to a phenotype of interest. So this is going beyond the alpha diversity measures and focusing more on the beta diversity measures of the microbiome. And then to be more specific, people are interested in identifying microbial signatures of disease. And this is more for prognostic, diagnostic. And so that refers more from a statistical sense to what we call variable selection. And then there are other experiments you can do where you can measure the microbiome, but you can also measure the omics from the microbiome or from the host as well. So you can have, for example, meta-transcriptomics, meta-metabolomics, meta-proteomics. So those are the omics measured on the microbiome, on the microorganisms. And then you have all the normal omics from the hosts. So you can have also transcriptomics, proteomics, metabolomics, and so on. And really here, from a statistical perspective, we're more interested in data integration and understanding what is the relationship between all these different molecular levels. And I will put a note here that I haven't mentioned any causal mechanism. So all of this is still quite descriptive. And even more so, I will show you later when we talk about network inference. So microbiome data haven't had a lot of statistical methods developed because they are inherently challenging. So it took statisticians a long, long time to catch up. And just to list a few characteristics, they're sparse, so you have a large number of zeros, they're compositional, and they're multivariate. But in addition to this, they're highly variable. They have often a non-Gaussian distribution. You can use different types of sequencing technology. You can use 16S amplicon sequencing. You can use shotgun metagenomics, for example. There's different levels of analysis you can do to make sense of your data, so that can be also quite confusing. And also, we worked a lot in my team about the effect of batches on microbiome. So the microbiome is highly sensitive to any changes in the environment, including batch effect. So we've done quite a bit of work with now my postdoc Eva on managing batch effects. So just to show you an example of how the data look like, on the rows you have the different samples. So here they annotated with where they're coming from, and on the columns you have the different names of the bacteria and then the counts that you have. So this is just a 16S data set. Usually we work at the operational taxonomy unit level, OTU. But then for the interpretation, we might go one level up to, genes or species or order, depending on the information we have. So the problem you can see from those data is the large number of zeros. Sometimes it's just not there. Sometimes it's there for some samples, but not others. And it's been always quite challenging to work out if this excess of zeros is due to an actual absence of the bacteria, or if it's just because we haven't sequenced deep enough, or maybe there's some sequencing error. For example, this one here, there might be some issues going on here. And then if you just take one bacteria, the counts of one bacteria, and you plot this in a histogram to look at the density, you can see that it's highly zero inflated. So this is like, the very close to zero or zero counts, and you still have counts that are much higher than zero. The other problem we have is this codependency between bacteria or microorganisms. So in ecology, where a lot of ecological methods have been applied to microbiome because it seemed like a natural thing to do. But in ecology, usually when we look at a species, we don't really assume that it affects the presence of another species. So I'll just give you an example here of ladybirds, tigers. Whereas in the microbiome, they are codependent. And so if we measure the abundance of one species, it's usually relative to whatever else you have in that given sample. And another issue that it poses is that we don't have the data in absolute value in sequencing. So we always have to think in terms of relative abundance. So that goes to the third challenge, which is that microbiome data are compositional. So from a statistical term, it means that the data are described as proportions. And so the proportion of one bacteria is highly dependent on the proportions of the other bacteria. So the origin of why we have compositional data is, as I mentioned previously, it can be just a natural thing. That if you have one species, then you need to have the other species because they work in a symbiotic way. It could be that you have technical artifacts because we sequence a finite amount of reads. So whatever you've been sequencing here will affect those compositions. And it can also be a result of the data transformation we do. So some people do rarefaction, which is when they start with a small amount of material. And so they have a sequencing library size that changes drastically between one sample and the other sample. And so what they may choose to do is to rarefy their data set where they undersample their reads so that all the samples roughly have the same amount of reads. So they downsample the samples who have been heavily sequenced. Some other people also calculate proportions, mostly to deal with the difference in library size. And so that creates compositional data just because you're working with proportions. And so I guess the warning here is that most statistical methods will assume that your data are unbounded, which it means that the counts should be between 0 to infinity, whereas in proportion is between 0 and 1. And so a lot of the statistical methods are not directly applicable to microbiome data when you have proportions or when you have compositional data. And Pearson had already noted this back in the 1897, where he said, actually, this will lead to spurious correlations if you just apply classical statistical methods on compositional data. And so there's been a whole range of papers in the recent years mentioning that it would be a problem if we consider microbiome data as they are and not as compositional. But there are solutions, and some are very pragmatic. So that's one we use in Xomics, is that we use a transformation based on ratio and based on log ratio. So in Xomics, for example, we use standard log ratio transformation. It's just a way to transform the data where you have your proportions and then it's projected into a unbounded space. There are some other log ratio techniques, ALR, ILR. The only thing you need to be aware of is that when you use those transformations, you should just always keep in mind that you're working with ratios now. And so your interpretation will always be in terms of ratio. And ratios is one way to circumvent that issue of compositionality. And so some other people have used proportional or proposed proportional distance or compositional balances. So we covered some examples in this paper if you want to have a look. So that's my first part of my talk. I'm happy to take any questions if you have any.
Quelqu'un indique. Pardon. Non, je vois pas de main levée, rien. Donc ça va.
Okay. All right. So I continue on to longitudinal microbiome studies and the challenges associated here. So when we started to work on longitudinal microbiome data, we realized that there were 3 types of analytical objectives you could ask to this data. And I've been interested in longitudinal omics studies for a very long time, but I didn't have enough data or experiments back then to really, be able to propose any new methods in that field. It only came quite recently, and especially in microbiome, because it's quite cheap now and quite easy, to have fecal sample over time from participants. So this is where I thought, okay, we can start to work on this. And so we wrote a review on just, what are the challenges associated with longitudinal microbiome studies. And we found that there were 3 main analytical objectives. The first one is a simple differential abundance of the bacteria over time and between sample groups. So here I'm showing you different plots where the x-axis is time and the y-axis is the relative abundance of a bacteria. So each line here represents the same bacteria abundance, but for different individuals. And the individuals have been colored according to the group, so either the control or treatment. So when you do a differential abundance analysis, you're asking whether there's a difference for a particular bacteria, if there's a difference between control treatments, or if there's a difference between control treatment and time. So this is an example where there's no difference at all. This is an example where there's a difference between the 2 treatment groups, but there's no difference across time. This is an example where you have a difference across time, but no difference between groups. This is a difference between time and group. And this is an interaction effect. So this is when, depending on the treatment you have, you're going to have a different behavior of that bacteria across time. So this is, a lot of methods have been proposed there. They are usually zero inflated statistical models. We proposed to use linear mixed model splines, and I talk a little bit about this in a few slides. The other objective is to just cluster the microorganism across time. And basically here, the question is a bit more advanced. It's saying, well, if I can cluster my bacteria across time, maybe they have the same biological function, or maybe they produce the same product or a similar product, or maybe they work in symbiosis to produce that product. And so here, what you have is each line can be different bacteria across time again. But this time I averaged the relative abundance of those bacteria across several individuals. So here I completely removed the information about the individuals. We just assume here that they are homogeneous enough in their group and that we're focusing only on a particular group of people. And so it becomes just a clustering exercise once you average those profiles across those individuals. And so you can just use any clustering method. I guess the trick is how you average across those individuals. So in our work, we've used linear mixed model splines to do that. So this is what I will present a bit later. And the third objective is to model the associations between bacteria. So here, for example, each node is a bacteria, and I will put a link if I think there's a negative or positive association between them. And so there are different methods that have been proposed, not many, and we have been proposing LUPINE. So just to give you a little summary of the state of the art, there are many differential analysis methods. They are univariate. The only thing you need to remember is that depending on the model you use, they can be applied to either counts or continuous counts that have been normalized. So for this, there's heaps of stuff. For clustering analysis that is multivariate, it becomes a bit more tricky. Usually it requires a small number of time points, because when you go beyond 10 time points, usually the system can become quite unstable, or you can have a lot of variability across time and things that happen that are not quite regular. So that's tricky from a clustering perspective. And the third objective about network modeling, we found that there were not many approaches. Actually, there are different types of approaches. One would focus on, trying to model those networks across all the individuals. But quite a bit of work has been proposed to model those networks per individual. So that makes it quite tricky, because then you need to kind of find either consensus between those networks. And just remember, this is all across time. So yeah, if you're interested, you can have a look at this review that we wrote, where we gave a lot of examples of analysis. So I just want to focus a little bit on objective 2 before I move to LUPINE. So objective 2, just a reminder, is to identify correlated time profiles across time. But here we had a more generic approach where we thought, oh, we could do that also with all these different omics, because why not? And so the data here are, if you think about them, you have different blocks. So you have the time, and then you can have all these different omics. So for example, you can have metabolomics, microbiome, transcriptomics. The tricky bit is that sometimes you have missing values. You can have missing values per individual. You can have missing values in a given omic. So the individual is just because they just didn't come or they didn't provide the sample. But sometimes you can also have a full omic data set missing at a given time point, just because maybe it was too expensive or something. So here, to differentiate between a classical omic analysis, we have a time dimension. And so the data, you can consider them as sort of cubes. So the data in 3D, you have samples, variables, and time. And so what we proposed in order to cluster those profiles is to reduce the dimension of the data from 3D to 2D by using smoothing splines. And smoothing splines are quite useful here because they can also estimate missing data. And so we end up with a profile of all the omics across time. And as I said, they are collapsed or summarized across a certain group of individuals, not taking the average. Here we're going to take the splines to do that. And so in the end, and then we can apply different type of clustering methods here. We propose to apply partial least square regression or PCA, because PLS in particular can help you to identify correlated profiles across omics data sets. And so you end up with identifying different profiles. So here we have RNA, metabolomics, and microbiome profiles that have been clustered according to PLS. And you can see we find some quite common behavior across time. So this is some work that I've done with Antoine Baudin, who was doing his PhD at the University of Laval, and also Olivier Chapler from INRAE, Anthony. And I just want to show you how it looks when we use those smoothing splines. So basically, imagine here that I have the relative abundance of one bacteria across time for each individual. So each dot here is the value of that same bacteria for different individuals. And instead of dealing with this 3D data, I'm going to summarize the trajectory across time using a smoothing spline. And so basically, I just take the values on that line here, and there's only one value per time point. So smoothing splines are great because they can reduce the noise. You can see here you have a little bit of some sort of outliers. And also you can interpolate the time points. So for example, we have missing time points here. I can still interpolate and say this is the value here that I'm missing. But the smoothing splines themselves can also have some limitations. And one is that you have to choose a smoothing parameter. So if your parameter is too high, then you over smooth, which means that you're following the data too much. And so your trajectory is not going to be regular, it's going to be a bit bumpy. Or if you under smooth, then you may have just a straight line. And so we looked instead of using a framework called linear mixed model spline. It's basically a linear mixed model that fits splines. It doesn't require any smoothing parameter. It's a flexible approach because you can also do a linear mixed model analysis in there. So you can ask, you can do hypothesis testing, and you can ask, for each bacteria, is there an effect of time? Is there an effect of groups, and so on? So it has a bit more flexibility. So that's what we propose to use in the time omics framework. And so going now to the third objective about modeling microbial networks, what we're trying to do here, imagine that you have a table of counts of those bacteria, but also across time. And we want to identify a network between those bacteria. So we want to identify co-occurrent bacteria. And be able to identify changes in association across time, and also identify what are the important members of the microbial communities across time. And we also want to characterize the changes in associations when you do an intervention. So the challenges here is that most people have used correlation matrix, so Pearson, Spearman, and Kendall, and this has some limitations. So I'll just show you an example here. This is 2 taxa counts. And you can see if you use Pearson, Kendall, or Spearman, then you have quite a low correlation. So you would assume those taxa are not associated in any way. But if you add a bunch of zeros to those taxa, so imagine now I'm adding more individuals and those individuals do not have this taxa, then suddenly the correlation coefficient increases regardless of the measure you use. And so that's create a spurious result that is not a true result. So this comes to the concept of partial correlation. So usually this is what we assume is a true correlation, okay, the ground truth that we don't have. And you can see there's an arrow because in the ground truth, we have a directed network. If you just calculate a correlation, so for example, Pearson or Spearman, you may end up finding a high correlation between the nodes X and Y, even though there's no correlation. And that's because they are linked via Z. And so there's this concept of partial correlation, which basically would not detect that spurious correlation because it's conditional on Z. So the idea of partial correlation is that we try to estimate the association between 2 nodes while taking into account whatever else is happening in this system. So that's really the idea of LUPINE that we're going to use now. So before I move on to LUPINE, I just wanted to ask if you have any questions.
Est-ce qu'il y a des questions? Alors, je vois pas de main levée. Moi, j'en ai peut-être une, mais j'ai peut-être raté une information sur quand tu fais alors je vais appeler ça l'imputation des données manquantes.
Oui, avec
Quand
les
il te
spins.
manque un jeu de données,
Oui.
je suis pas sûre.
En fait, tu peux imaginer ici que, okay, so the question is if you're missing one omic at a given time point, you can imagine here that I have a complete, so at that time, for example, I have 0 information on my microbiome dataset, but I have the information before and after for a given omic variable. And so you just take that value here.
OK.
So yeah, it's interpolation. And yeah, so in that paper here, this one, it was interesting because we had, I think the metabolomics, we didn't have many time points. We had less time points on the metabolomics data that we had on the microbiome because metabolomics data are a bit more fiddly and expensive. So we may have had like 3 time points for metabolomics. And then we had like 5 time points for microbiome. But we had the start and the end and the middle. And so we could start to smooth those, the splines of the metabolites when we were missing some time points there and there. Just fill it with the spline inference.
Okay, Natalie, you have a question?
Yes, on this slide. Perfect. Yes, I was wondering, how are you doing to align scales of the different omics in the plot that we see? You see that abundance are quite similar between RNA and metabolites and so on. And how is it related to the compositional nature of the data?
Yeah, so there's 2 questions in there. The first one is, just a change of scale. So you have to remember now that we have 2D data after spline modeling. So you have the number of time points and the number of variables. And we put that into a PLS or a PCA. And so what we do in PLS or PCA is we center and scale on the variables, on the columns. So we always do that in PLS. So this is a PLS result, by the way, because we have different omics. We always do that in PLS to account a little bit for the difference in scale.
Yeah, but
and
across times. So you scale, but not.
Yeah, so across all
times.
time points.
Okay.
Yeah. So that's one way, and then the PLS, will handle the fact that you have different omics. So that's one question. The second question about the compositional data, I don't think we have fully addressed. What we do here for the microbiome is that we do a CLR transformation, and our rationale or argument would be to say, well, look, it's really hard because, you have it across time as well with different proportions. And so we think that just using a CLR transformation will help to transform the data outside the simplex and be like in a normal thing. It's not perfect. And in that paper here, we looked at different ways of validating the results. That way, we looked at proportional distances and stuff to say, well, look, it's not too bad actually. But there's still, quite some issues, I think, when you think about the longitudinal microbiome. And we actually, in that paper here, we really mention what would be the issue when you have compositional data that you measure across time, because of course the library size is not the same for a given individual at a given across time. You may have a different library sizes across time for that single individual. Yeah.
Okay, thank you.
Yeah, and I think the approach of Lupine also addresses a part of that.
Thank you. Any other question? I don't think so. You can go.
Okay. All right, so now I'm going to talk about Lupine. I often say in my lab, we try to be very innovative about how we name our method, but then I completely forget and I think everybody forgets what the acronym is, and it doesn't really matter. So Lupine is a method based on PLS. for network inference. And the idea of Lupine is how we can leverage the past information that we have to model a network at a given time point. So I'm going to talk about partial correlation. And because we use PLS, we're also going to use low dimensional data representation to do that. So this is the work that has been done by my postdoc, Sarita, who's still working with me at the University of Melbourne. And we have the manuscript in revision. So the idea of Lupine is how do we estimate partial correlations between two taxa? So this is how the dataset is looking like for a given time point. Just going to start with a simple example here. We have our samples in rows, our counts of taxa, and each taxa or each species is in a column. And what we want is we want to estimate the association or the correlation between taxa I and taxa J. As I told you before, if you just do a normal classical correlation, it's likely to be spurious because there's all the green taxa that we are also measuring. So one way is to do a conditional analysis, similar to what I was explaining before when I showed you, the partial correlation between 3 nodes, except that here we have many, many nodes. And so one way to do this is to say, okay, let's remove taxa I and J for now. And so we just end up with the green taxa. And in order to do this conditional analysis, which is too hard when you have way too many taxa, we're going to summarize the green taxa by a component. So you can do that with principal component analysis. You apply PCA on this data set, and you end up with one component. And you use that component as a summary of all the information you have on this green data set. So you end up with this summary of the green taxa and then the taxa of interest, I and J. And we also have the information about the library size because I told you, because of those compositional issues and stuff, we have different library size often for the individuals. And once we have this, then we can just fit a simple conditional regression model where we model the log counts of each taxa, I and J. So it's similar models, just using a regression, a linear regression, where we include the green taxa summary here. We also include the library size. So that will probably answer Natalie's question about, how you handle different compositional issues more in the terms of the library size by putting it into that model. So once we've done that conditional analysis, what remains is what we think is basically the information that remains after you remove the library effect and after you remove the information about the other taxa. So those are the residuals. And basically from there, you calculate the partial correlation by calculating just the correlation between the residuals of taxa I and taxa J. And according to a correlation test, if you say, well, that correlation is actually quite high, then we say, okay, there is an association between those 2 nodes, I and J. So that's really the idea. The idea is to say, well, in order to estimate this partial correlation, I'm just going to summarize all the information I have on the green taxa using a simple dimension reduction technique such as principal component analysis. And you have to do that for every pair of taxa. Usually in a microbiome dataset, you don't have that many species. You may have up to 500. And in the paper, we describe how we can speed up the results by using some, approximations to calculate those pairwise associations between taxa. So this is just for a single time point. And then we can generalize this idea for more than one time point. And so for that, we're going to use PLS models. PLS has been, well, in my lab, we've designed it so that you can integrate 2 omics data sets. So you could imagine here you have one omic data set and another omic data set. And from the PLS, what you get is a component that summarizes the first omic data set, and then you have another component that summarizes the second omic data set. But those two components are maximally covariant, so they're correlated. And so this is how you extract common information between the two data sets. In this context, where we want to estimate the partial correlation and we still want to use those components, we have each omic data set is replaced by the different time points of your microbiome study. So for example, if I'm interested in estimating the association of taxa I and J at time point 2, the other omic is going to be the same data set, but at time point 1, so in the past. And I'm going to do the same. I want to estimate the partial correlation between I and J at time point 2. I'm going to extract first the green taxa that I'm not interested in. I'm going to summarize them, this time using the PLS. So you have two components. And then I'm going to include those components, the last component here, into the conditional regression analysis. And basically the idea is to say, well, I only use this one because this one is also correlated with the past. So it's actually already including all the information about the other taxa from the past in order to estimate my partial correlation. And then you can go, and of course you have the library size as well. So it's the same, framework as before. And then you can generalize that to multiple time points where you have, and for that, we use a multi-block PLS. The idea is the same, but there you may have different time points. So you have 3 to 5 time points. So here, for example, I might be interested in estimating the association between two taxa at time point 5. And I'm going to have all the components from the past, from time point 1, 2, 3, 4. And I'm going to extract the components from the block PLS to include all the past information there. And then once you have that, you just do the same. So you extract the residuals, and then you can estimate the partial correlation. And then you can say, yes, based on that modeling, those two taxa are associated at a given time point, but I have taken into account the information I had from the past time points. So we were interested also to measure how the quality of a network. So we use different measures. One is just looking at the distance between network topologies, so comparing two networks. We visualize this with multi-dimensional scaling. We also were interested in understanding the inference of the nodes, the inference of the taxa. I'll show you an example later. We also can visualize that using PCA. And also, we also did develop some correlation tests so that you can tell us how similar two networks are. So that's quite interesting when you have, because we're going to model a network at a given time point while taking into account the information from the past time points. So we might be interested to say, well, from time point 2 to 3, is there much difference between those networks of taxa? I'm going to skip the details about the simulation studies because, it's not quite exciting. But basically, we showed that our method had a better performance than other methods, such as SparseCC and SpeakEasy. SparseCC and SpeakEasy are also network inference methods for microbiome data. SparseCC is just based on correlation, and SpeakEasy is based on the concept of partial correlation, but none of them have actually been developed for longitudinal data. So they can only focus on one time point. And we also compared our method LUPINE with the LUPINE at a single time point in a longitudinal setting to see whether there was any gain in actually including past information in the partial correlation estimation. And we show that it brings much more robust patterns when you take into account the time. So now I'm going to move on to some examples and then happy to take some questions. I have 3 examples here. The first one I want to show you is just a high-fat, high-sugar diet study in mice. So here we have 24 mice, 22 or 24 mice per group, a normal diet or a high-fat, high-sugar diet. We're measuring the fecal samples of the mice at 4 time points from day 0 to day 7. And this is just to show you how the network look like when you visualize them. So after we infer the partial correlation between all the taxa, so all the taxa are represented with a dot. You can see on a normal diet, the networks don't change that much. There is a little bit of variability. Whereas in the high-fat, high-sugar diet, you can see that there is quite a few changes around specific species. So this is just one way of visualizing the results on a very, well-powered but small number of time points case study. The other one that we looked at is another study where you only have 9 mice. And we're looking at antibiotic resistance. So these are data from my collaborator at the University of Melbourne. We have a naive phase where basically nothing happens. So we have 3 time points, 4 time points, and then we give the mice an antibiotic. So antibiotic, as you know, will completely wipe out their microbiome. Then there's a wean-off period where nothing happens, and then we colonize the mice with vancomycin-resistant Enterococcus faecium, and we see whether their microbiome then recover to a naive or a normal microbiome. So this is just one group of mice. And you can see the networks, how they evolve across time. So you can see at the beginning it's quite dense because this is a naive phase, but as soon as you apply the antibiotic phase, those networks change drastically. And what's interesting is that when you have the colonization phase, then the microbiome starts to recover slowly. So this is what you describe here. Clostridalis reduces until day 14 and then starts again. Bacteroidealis appears to recover a bit earlier. If we look at the network topology, we can see the same. So this is visualized here in this MDS plot. This is a naive phase here. This is the antibiotic phase, and this is the colonization phase here. So you can see that we see a very strong difference between the naive phase and the antibiotic. And then we can see that the network, slowly, change across time during the colonization phase. And this is just a plot to show you the p-values of the Mantle test to show you whether there's a strong correlation between the networks. So when the p-value is quite low, it means that the networks are very similar to each other. And we expect to see this. So during the naive phase, the networks are very similar to each other, and then they change. And so there's the antibiotic phase where, again, the networks are similar, and then they slowly evolve to the colonization phase. So this is an example where we have a large number of time points, but a very small number of mice. And finally, the third example is in humans. So, much higher variability, small number of time points. We have 2 groups. We have a group of individuals who have a plant diet and a group of individuals who have an animal diet. And then there's a wean-off period. So if you just visualize this with the MDS plot on the similarities between the networks that we inferred. So each dot here is a network that we model per day. What we find is that for the plant diet, the baseline, the plant and the wean-off are very similar, which suggests that the bacteria are more or less the same. And then if you change to a plant diet, it's not going to affect your microbiome drastically. So they all remain more or less the same. Whereas for the animal diet, what is interesting is that this is the baseline, and this is when people start to have an animal diet. And then the other dots in orange are when there's a wean-off period. So when you actually go back to your normal diet, and you can see that there is a major shift in the in your microbiome when you switch only to animal diet for a little bit of time, and then when you wean off, it goes back to where you were. I mentioned the IV measure here. Sorry, I'm just going to show you how it looks into some of the networks. We couldn't show all the 11 time points here, but just to show you, how the networks can change across time. We just selected 3 days. And you can see we just highlighted very specific bacteria that were present in either the plant diet or the animal diet when people had that diet. And yeah, just to go back, one other question you can ask in this network inference is, what are the bacteria that have an influence in those networks? And they can be represented with this IV score. So for each bacteria, which are in columns, and for each time points, you can have a score, and the score is going to tell you whether this bacteria is influential in your network or not. And so, for example, there are some specific bacteria that are always there, regardless of the diet you have, regardless of the wean-off or the naive phase across the plant-based and the animal, which means that those bacteria are not affected by the diet or any daily variation. And then you can identify some bacteria that are only here at a very specific time point and together. So I just wanted to show you, 3 examples, some in mice with different groups, some in mice, but with a small number of mice and only one group. And then in humans with 11 time points and a small number of individuals per group. In the paper, we also have, on the request of the third reviewer, also included a very large cohort in humans. And just to summarize, this is a sequential inference approach for microbial networks in longitudinal experiments. What we were interested in is whether we can detect association or stability of associations over time and as a response to intervention. I'll show you how we use PCA and PLS to reduce the dimension of the data so that we can estimate the partial correlation and infer those networks. And we use a bunch of metrics to also, give more insights into those networks. This method has been designed specifically for small sample sizes. And by small, I mean about 10 per group, because you need to remember that we are calculating a partial correlation or a correlation between different taxa. So the package at the moment is available on GitHub. And we hope to put it in mixomics very soon. And just so you know, we are running our next online mixomics workshop in about 3 weeks. Thank you.
Merci, Kimane. Is there any question?
Natalie has one.
Yes, I have a question about the fact to use PCA as a previous step to compute partial correlation compared to the approach of Munchausen, which directly puts all the variables with a lasso or elastic neck penalty into a linear regression framework. Is it more efficient, or?
It's definitely more efficient.
Yeah.
I would say,
from a computational point of view.
Yeah, and also because, I mean, we have like 500 or so taxa to put in. There's also the issue, I mean, can be circumvented by TLR transformation, where you say, okay, it's data Gaussian distribution. But I think for us, the PCA one was more like it's an option for a single time point, but we were much more interested in multiple time points. And this is where the PLS was, much more attractive. And so we added the PCA afterwards. More as or sometimes we also say in the paper, look, if you have a very strong intervention where really your networks are not linked between time points because it's either far in time or just very, very different, then it's probably better for you to use a single time point approach.
Okay, thank you.
Did you test Lupin with other type of omic data?
No, but I think it can be generalized easily because, you know, it's just a PTA PLS approach. I think we did it first for microbiome data because it's a natural way to think about microbiome data. You know, we always think in terms of networks of bacteria. Whereas I think in omics, I mean, we're also interested in gene networks and protein networks, but there's a lot of things that have been done, you know, in that area. I don't know how much has been done for longitudinal analysis, but definitely for single time points, there was so many, you know, approaches there. What would be interesting is to extend that for multi-omics, you know, and look at the bipartite, what we call bipartite network, so the network between a gene and a protein. Yeah.
Okay, thank you.